All right, I asked in the chat box. I need to find out. You know, I have a feeling that the chat box in the video of me doesn't show up when there's the recordings. When you view the recordings. Anyway, so in the chat box, I asked, "Where's Waldo?" So this is where we left off with the prokaryotes last time we were together. We were talking about how we can separate them in very basic ways. Remember, these things are really small, so anything that can help us separate them that's real obvious is going to be very beneficial. And one of the things we can use is the color they are after we do a Gram stain. And that's important in a few levels. Obviously, it's important in epidemiology and disease and medicine. Hi, Jesse. How are you today? Titania, how are you? But it's also important, of course, from a biological point of view. No kidding. So there's no recording, or there's no video, and there's no chat. Doggone it. Well, we're switching to something anyway. Good. So this slide that we ended up with was the results of Gram staining on bacteria. You remember what I said? That's one of the very big ways we can decide the two different types. And we're going to talk about some different, more specifically about different prokaryotes today, some Gram positives and some Gram negatives. And remember, that's the basis of the stain. If you're Gram positive, it's purple. Gram negative, you're going to be red. But beyond just that, remember why it's that way. Remember, there's something very significantly different about bacteria that have Gram positive, or that are Gram positive or Gram negative, about their peptidoglycan layer. That layer that's, okay, how many slides? I got to go back just two, right? I'll do that. See, the ones on the left, the Gram positive, the layer is much thicker. The ones on the right have that double layer. Yeah, that's right, but it's a peptidoglycan that we're focusing on. It's much thinner in Gram negative. Remember, the alcohol in the Gram stain shrinks the peptidoglycan layer. And since the Gram positive ones have a thicker one, that shrink wrap, think of like a shrink wrap around produce or whatever, the thicker the shrink wrap is to start off with, the thicker the shrink wrap, why did I pick that word, is going to be at the end. You see what I mean? The plastic that starts out that's thicker is going to end up thinner, yeah, when you shrink wrap it, but not as thin as something that's very thin. So I guess when you make that stuff, you got to balance that out. Makes sense, right? Oops. Bailed out of here. So the one on the right-hand side has the much thinner layer. So, of course, what happens then is the alcohol not only shrinks the peptidoglycan, but in the Gram negative, it washes the purple dye back out. And then remember, we come back later on and we dye it red only so we could see it. We don't dye it red for any reason other than so we can tell it from the Gram positive. That's it. Hi, Janae. How are you? And the purple ones dye too with red. It's just to think that since they're purple, you can't see the red. They're already purple. Good. So we were just going back over the basis of Gram positive and Gram negative. So looking in this slide, very easy. It's one of the easiest things to do in microbiology. But like on an exam, for example, I wouldn't necessarily just stick with, hey, tell me which ones are Gram positive and Gram negative. I might say, okay, what else do we know about the Gram positives and Gram negatives that makes them different? Other than the fact that they turn out a different color, can you relate that to the form and to the organismal structure, right, to that peptidoglycan layer? So we're still kind of talking about the outside of prokaryotic organisms, because there's a lot going on the outside, because these kind of critters have a lot of not that any of us don't, have a lot of interaction with the organisms around them. In many cases, it's not necessarily good. Like in this picture here, this is Streptococcus, obviously the organism that causes strep throat, sticking to a tonsil cell. So this would obviously be the beginning of strep throat. And you can see there's the bacterial cell wall, and that's going to be where the peptidoglycan and the phospholipids and all that are. But in addition to that cell wall, there's also in many bacteria there's a capsule that's around there, right? So and oftentimes what that capsule is, that it's a surrounding, and it might protect the thing from drying out, or it might, in addition to that, hide that bacterial cell from an immune system in a host. You remember back in Bio-1, glycolipids, glycoproteins. Now that I know this doesn't show up on the recordings, anyway. Glycoproteins, they were on the outside of the cells, of cell membrane, sticking out into the environment. Do y'all remember what the function of glycolipids and glycoproteins is? You remember that? I'll give you a little hint. The structure of glycolipids and the glycoproteins is variable. It's genetic. It's based on you. Your genes decide what those structures of those things sticking out, not in, but out of the cells. Oh, come on, I can't believe that, because I think that's one of the neatest things when you kind of learn, like, how does my immune system, with these white blood cells and these other parts of it, as it makes its way through my body, how does it know things to attack and things not to attack? I mean, it can attack living things. It attacks bacteria, but bacteria are cells. How come it doesn't attack my own cells? And yeah, I know, autoimmune disorders aside, why doesn't my immune system kill my good healthy cells versus just the foreign invaders, the pathogens, whatever you want to call it, the cells that quite clearly the immune system has figured out aren't supposed to be there, right? How does it know that? They can't talk. One cell doesn't say, "Excuse me, can I see your ID, sir?" Right? So what is it? It's really sort of a repeat of what I talked about the other day. We taught down in class when we were doing enzyme. I was using her as an example, enzyme and substrate, right? The things happen in chemistry, things happen in biology and metabolism for actually a very simple reason. Enzymes and substrates get together because they fit. It's all about structure. Remember, it's all about shape. Well, by chance brings them close together, right? By chance. But the final thing about whether it's going to be the substrate in the enzyme or something else is going to try to bind there is the cell interaction. The cell interaction, or just in the case of the enzyme and the substrate, the molecules fitting. So those glycolipids and glycoproteins that are on the outside are about shape. So your immune system, how it knows a cell is yours or is a cell from the outside, like a bacterial cell that might want to cause you harm, is it checks the glycolipids. It feels, if you will, right? It doesn't have fingers. But it goes, oh, yes, these are the correct shape. And that's how it knows. It doesn't have a brain. But you see what I mean, right? It's got to have, it's all about, it's kind of like the example I use in my Bio-1. The key to my truck only only opens my truck. It doesn't know anything. Is because it's shaped right. It's the same thing with enzymes and substrates. It's the same thing with immune system knowing that this the thing is either something that's host or foreign or some sort of and because it's foreign doesn't mean it's necessarily disease causing, but many times it is. So you want to take your obviously now those of you who have allergies, you realize that it's all that's your immune system going crazy. That's your immune system reacting to things like pollen and recognizing those as foreign, but pollen's not going to kill you. But your immune system thinks, oh my gosh, you're going to get this out of here. It's actually your immune system kind of haywired. It's interesting, right? So one of the things we can have besides the cell wall, of course, is this slime layer. The slime layer can help hide them, or a capsule can help hide them. Slime layers tend to be a little less dense, and slime layers tend to be a little better at doing this thing of adherence, right? Sticking the bacterial cell, the streptococcus, to the tonsil cell. Because and again, how did those come together? What Tatiana said, they come together by chance. But with that slime layer, that you stick to it, and then you can start reproducing and give you strep throat. And we said these guys were good. Now, speaking of not so good, anybody know what disease this one causes? The scientific name of it is in the upper left. Notice it's a bacillus too. It's a rod, right? It's a rod shaped, just like Staphylococcus and Streptococcus. That's the shape as well as the name of it. But in this one, other than the shape, you might get a clue from the species name. There's a horrible disease out there that causes massive pandemics, and it's actually, for what been turned into a weapon, a military weapon in some cases. Right after 9/11, there was a scare where people were receiving spores, and that's what you're looking at here is an endospore, because one of the other modifications that bacterial cells can do is they can actually produce endospores. An endospore is like a deep sleep, if they don't sleep, but a deep sleep for bacterial cells, and they can survive for a long period of time as endospores. In some cases, depending on species, I don't think I'm off, but there's some that are known to survive maybe decades like this. And an endospore is something that you might do if you're an organism in which the environment changes and it's not going your way. You know what I mean? It's not going your way the way the environment is. So what I'm going to do is I'm going to kind of go into this deep sleep, something maybe hibernation's are really the wrong word, but at least gives you an idea. And I'm going to wait for the environment to change, because with Darwin, the environment controls organisms' survival rate, right? But one of the things organisms can do, like these guys here, these organisms, not guys, is they can form these very resistant endospores. Sometimes we just call them spores. Endo means they're inside. And the outside of that organism there, no matter what happens to it, that endospore will stay resistant. And then 50, 100 years, when it finds itself in the right environment, in the case of this particular organism, with the name up there, it's going to be a moist, warm environment. And if that's what kind of environment it finds itself in, then it's going to germinate and become a living thing. Now, when they're a spore, are they dead? No, they're not dead, because dead can't come back to life. Are they alive? No, they're, yes, yet kinda, barely, sort of. Kind of like bears hibernating. The bear's not dead, right? Just really in a deep sleep, kind of like the way my son used to sleep. Nobody figured out what disease this organism causes? Bacillus anthracis. If you just take off the CIS at the end of there and put an X, anthrax. This is the disease anthrax. This causes anthrax. What's anthrax? Anybody know what anthrax is? I heard somebody click in here. It can, yeah. It can cause death. Okay, anthrax is an infection caused is the infection that's caused by this disease, and you can inhale it. It gets on your moist membranes, and it can cause all kinds of nasty stuff. We can get contact through infected animals, inhaling the spores. That's where the weapons part comes in. But anyway, it can be really, well, it can be fatal. It's not always fatal, but it can be fatal. And so, one of the things, like I said, they've been using for in weapons research is if you dump this all over the enemy as spores, then they inhale it, and the things germinate, then you can have this particularly life-threatening disease. There are cases many times where people live through anthrax, but anthrax, especially if it goes untreated, can cause disease. Yeah, all kinds of stuff. Skin disease can look like the what's the flesh-eating bacteria kind of thing too. This isn't the flesh-eating. All right, another thing that they can do, and this can help them with adherence. One thing is they can produce these little extensions called fimbriae. And the fimbriae, for those organisms like that Streptococcus on the tonsil cell, similar to that, it can use these fimbriae to actually attach themselves. In many cases, if you're a parasite again, or you need a host. And in some cases, these fimbriae can actually grow between two of them, or in this particular case in the picture, just one and another one. And that fimbriae, since it's a conduit or a tube, it can act as an exchange for DNA. We call it a sex pili. These organisms don't really have its sex, obviously, in the way that we think about animals. But what they do is they do exchange DNA, and that's something I'm going to talk about in just a bit. But that has something to do with generating genetic diversity in these organisms. See, the thing that we always challenge ourselves with when we think about prokaryotes is they're single-celled, and when they reproduce, they do by cloning. And cloning makes exact copies of the DNA. So how do we get variability? And if there weren't, or we don't get variability, then of course that's going to be it's a good chance of extinction, because that's how variability that's how variability staves off extinction, right? Not everybody dies, or not everybody. Some of us have an advantage. Even some prokaryotes. All right, about half prokaryotes can move around. Some are pretty fast at scale rates of running somewhere in the vicinity of 90 miles an hour. So apparently some of those organisms have someplace they need to be pretty quick. Methods, the flagella is the most common, that little spinny thing, just like eukaryotic cells. Now, it is a flagella, but I'm going to show you, let me show you right now. Let's take a look at the flagella in a prokaryotic cell and a eukaryotic cell. It's a really cool story. All right, up in the upper left is the flagella of the prokaryotic cell. All right, now I'm not real concerned about you knowing the structure. You can see this is obviously, we're looking at a gram-negative. That's a thin peptidoglycan layer. It's got the double layer. You can see the little rotors. You can see the flagella, and you can even see it in an electron microscope picture. What's cool about it is you can see all the intricacies of the detail, man. This is neat. It's a little motor. People who are into nanotechnology get fascinated by this stuff. And the flagella spins. It spins around, and that's what propels your organism. All right, very good. Notice in the red arrows, the protons, they're flowing. If you remember back to Bio 1 and photosynthesis and respiration, you were introduced to a particle called ATP synthase. It almost looked like a lollipop shape. And you remember protons flowing through there would help generate ATP. Strike a bell? Ring a bell? Kinda? Sorta? Well, notice right here what we have is protons flowing through something, getting something to spin. That's what the protons were doing when they went through ATP. I don't know if you remember, but ATP was being generated there because the protons were spinning something. They were turning something. The actual proton flowing through microscopically, a proton going through was making this thing turn around, and it was making ATP. Now look, same thing's working here. But what we get is something spinning around, but it's not making ATP. It's actually a flagella. Now, I label that as an excitation. I don't know if it's an excitation, but maybe it was. Maybe initially protons flowing through something was used to spin a flagella, and later on it became beneficial for making a bunch of ATP. Because you remember, you don't get a lot of ATP, or most of it, until you get into the mitochondria, in respiration anyway, right? And you don't get into the mitochondria unless you're doing aerobic respiration. That would have come presumably after critters with prokaryotes had these spinning flagellal cells. Flagellal structures. All right, now down on the lower right, what I'm trying to show you, and again, if you remember in eukaryotic cells, the way the flagella moved was microtubules, that cytoskeleton stuff, would move back and forth across each other. Like there's a bunch of them. If you imagine my fingers are a bunch. And when they did, they would cause one side of the flagella to be shorter, longer. So if you imagine see? If they're moving up and down like that, so that's what caused the flagella to move in the eukaryotic cell. So prokaryotic cells and eukaryotic cells both have flagella. Flagella is a little wispy thing. You usually have, few of them. They're mini cilia, typically 1, 2, maybe a half dozen flagella or flagellum. Both work the same way. They move around and they cause the cell to move. Very different structure, though. Very different. The way the eukaryotic cell flagella is constructed, the way it works, is very different than the way the prokaryotic one. So now, what fundamental principle in evolution did we discuss that this is reflecting, the prokaryotic and eukaryotic cells flagella? In other words, during our concept of evolution or our discussion about evolution, we talked about how we recognize relationships between organisms by how they look, by similar structures that they both possess. But we always have to be careful because sometimes organisms might look alike, but it doesn't mean that they're related to one another. Remember marsupials and placental mammals? So that's kind of what's going on here. These things basically do the same job, but their structure's not very similar at all, at least when it comes down to the actual mechanism, how it works. So in evolution, what would we call that? How would we explain that both these critters have flagella? A eukaryotic protist and some prokaryotic bacterial cell. Now, I know you. Hopefully, you're thinking anyway that environment is the right answer, and of course it is environment. This would convey an advantage, but what I'm trying to say is in evolution, what do we call that when organisms? Yeah, isn't this kind of neat? Yeah, that's right, Tanya. Convergent evolution, down at the very basic level. We can see examples of organisms, single-celled eukaryotes, prokaryotes with flagella, coming up with similar answers to this common problem, the environment. I mean, to me, that's just amazing to think. And of course, we talked about it in dolphins and sharks and up at that level. But here we go right down to it happens even at if you want to be, again, apologies to all prokaryotes out there that are listening, even at the simplest level, even at the simplest eukaryotic level down at those protozoans, we have this idea of a flagella both responding to a similar environment. Got to have this thing to move you around, and moving around is beneficial, but just doing it differently. Okay? I got to go back. I got to finish this one up. How else do they move around? All right, the spiral ones. All right, hold on. Cooper, come on, in or out? All right, the spiral ones, the spirillum that we were talking about, remember the three shapes? They move by these filaments, what they call helical filaments. Helical filaments basically, if this is the spiral-shaped critter, they run this way. They run down. They're like long fibers on either side of the organism, and they basically pull back and forth, and it kind of wiggles. Right, it's not very efficient, but it does a good enough job. It's funny though, even in some of the worms we talk about, they kind of move in a similar sort of fashion as even the spiral prokaryotes do. Some bacteria have slimy threads, like we talked about, which they use to stick to tonsil cells. Some can actually use those as a way to actually sort of pull themselves along. It seems kind of strange, but they would very slowly. You can imagine this is fast. They would secrete slime out in front of them, and then they would be able to pull that slime in, and they would pull themselves along. Like I said, these guys aren't actually booking too fast, are they? They're just moving pretty quick, very, very slowly. But it is motility. Nobody says you got to go fast. You just got to get there. So if you have an ability to move, then you might exhibit a characteristic we know as taxis. All right, hence the word taxi. Well, I feel like that Greek guy in that movie. Give me any Greek word. These are pretty quick or easy to figure out, right? Taxis is movement. So if it's chemo, it's chemicals. Photo, it's light. And geo, it's earth. So among organisms in general, if you think about plants have phototaxis. They respond to light. Organisms, prokaryotic organisms could obviously chemo. We have chemotaxis. If you smelled something good and you were hungry, you would follow your nose towards it. That's smelling something. That's what chemotaxis is. Bacterial cells have that, so they might sense environmental chemicals, especially if they need ones for metabolism, or if they have certain environmental chemicals they want to stay away from. So you can have positive and negative taxis, especially if you're photosynthetic. Light would be important. Magnetic, that's kind of cool. Now, some people argue, well, not argue, but human beings don't really have magnetic sense, but like some birds, we believe, do. and some migrating fish, migrating birds too. And those magnetic particles in their cells actually align up with the fields, and they're able to sense that, which is kind of cool, and they can tell north. That's how, like a lot of birds, what is it, the tern, Arctic tern, or it's one of them that it migrates from the north to the South Pole every year. A lot of it over open ocean. So, boy, unless they got some really good, I wonder if just one of them has an iPhone, though. Well, I don't know what they did before iPhones, but they think they might be able to use the stars, and maybe in addition, I know when I was doing my research on eels, I found out that there are some eels they think might have the ability to sense magnetic fields. And so they can use that. Magnetic fields are not just north-south. They vary depending on where you are on Earth. There's minor differences. So like if those salmon salmon would be chemotaxis too, right? They smell the chemical where they were born. That's how they know exactly the little stream to get back to. They are in the ocean when they're living as adults, and time to reproduce, they swim by the coast. They go, that's home. Little tiny bits of chemical, and they can actually make it all the way up to where they were born. They're very sensitive to it. But you could do the same thing if you're sensitive to magnetic fields. And magnetic fields would help you, especially in a lake, knowing top from bottom. Right in the bottom, why would you need to know? That's where the nutrients are. So you want to get to the bottom. So photosynthetic ones might want to move to the light, or photosynthetic ones might be also geotaxis. So during the day when the sun's out, they rise, and when the sun goes down, they go down to the bottom to soak up some of those nutrients at the bottom of a lake. Magnetic fields change that way too. So it's not just north-south. Bottom of the lake, is the magnetic field would be different than the one at the top. I want to say stronger, but I don't think that's right. Gravity is stronger at the bottom of the lake, just a little bit. Organization. Well, not a lot, right? We talked about this. There's not a lot of organization of these guys. Not to say that they're disorganized, because that's a characteristic of life. But what we mean by that is not a lot of sophistication, right? They don't have a nucleus. There's none of that internal compartmentalization that the eukaryotes were known for. Remember why that was so important too? Because that means that the eukaryotes, eukaryotic cells, could do more advanced processing and safer processing. Like, for example, remember the peroxisome? You might not, but that was an organelle that processes hydrogen peroxide in your cells. Highly dangerous, It's like a toxic waste site there. But because it's compartmentalized in a membrane, everything inside of it, membrane within membranes and all that stuff, you can do that kind of job, right? Maybe you got an appreciation for it, but if not, hopefully I can add or actually provide an appreciation. The reactions you talked about in Bio 1, the photosynthesis and the respiration, specifically the respiration, the mitochondria part, and especially in photosynthesis, the light reactions part, the part that happened on the membranes. One was in the mitochondrial membrane, one was on the thylakoid membrane. See, you see what they're listed here, right? Those reactions only work, right? In spite of all the complications and all the different reaction part, you have to remember NAD and FAD, and this molecule becomes this molecule and all this kind of stuff. Those reactions only work if, and certainly in the case of aerobic respiration, the last part, if you can move protons across the membrane. Remember, in the mitochondria, we can only get ATP if we get protons across the membrane. And we can do that with proton pumps to get their energy from the electrons we get from food. But you got to have a membrane. In the thylakoid, ATP can only be made because you're moving protons across the membrane. You see that? And throughout your studies, especially if you go on an A&P, you learn it's not just protons across membranes. There's all kinds of things that living things separate across with membranes. Now, why would they want to separate? Well, like in the case of protons, if I can force more protons on one side than the other, I can cause Mother Nature diffusion, simple diffusion, the rock rolls downhill by itself, to flow back through ATP synthase, causing this thing to spin, making ATP. You remember ATP synthase is the same in the mitochondria as on the thylakoid membrane. Mother Nature doesn't reinvent the wheel sometimes, right? So what you're seeing in those pictures is an aerobic prokaryote or a photosynthetic prokaryote, but what I want you to kind of take away from all this is even though these guys don't have mitochondria, they were mitochondria, and they don't have chloroplasts, and we think, of course, with the endosymbiotic theory that they were chloroplasts, they still got membranes because they still need to separate things, right? They're separating protons, or they might be separating other things. That's very important. That's why we focus so much in those reactions on the actual membrane. What's happening there? Okay. So prokaryotes don't have organelles, but they can still make membranes and do that, move things across. Right? Now, okay, they're not sophisticated. They have a very simple genome. A genome is, well, it's variable what genome means. A genome is a collection of genetic material. So you are a genome. Us as humans are a genome. A population of squirrels living around downtown Orlando is a genome. You see what I mean? A collective sense of genetic information. That's shared and all that stuff like we were talking about earlier in evolution. Prokaryotes are relatively simple in their genome. Within a prokaryote, they, have one chromosome, right? We have several. We have 23 pairs in the case of human beings. They have in case of prokaryotes, about 1,000th as much DNA as the eukaryotes in a single loop, in not even a nucleus, right? Or an anuclear. It's a nucleoid region. So there's not a lot of genetic variability within a prokaryote. There's not a lot of genetic variability within a bunch of prokaryotes. But they got one thing that really helps them survive, folks, and that's the fact they can reproduce very fast. And in spite of the fact that they're making clones, as we're going to get into in just a moment, they are able to generate genetic diversity. All right, another thing that these organisms have, and this is really a testament, or at least a hallmark or a central element of their ability to survive, is they have these accessory pieces of DNA called plasmids. See the really cool picture on the right? That's a prokaryotic cell on the bottom, and all that spaghetti stuff sticking out is its chromosome. If you undid that whole thing laid chromosome, it would end up as one loop. Right? It's amazing if you think about how much genetic material, and that's just a prokaryote. But we're looking at the littler loops. See the one, there's a couple of them, plasmids. Those are accessory loops of DNA, usually smaller, well, not usually, but they're smaller pieces of DNA with fewer genes on them, and oftentimes those genes are the problem that we have as human beings, because oftentimes plasmids will have genes on them for things like antibiotic resistance or special contingencies like different types of diets, different types of energy sources, different types of sugars that they can use. Now, the antibiotic resistance within an organism with its plasmid unto itself isn't really too scary. That's one critter, right? That's one organism with one plasmid that says, hey, I don't care about antibiotics. But now the plasmids can replicate. They can replicate independent of what the chromosome is doing. And then, and this is kind of the freaky part, the organism that has the plasmid can give it to another organism. So we call this horizontal gene transfer. It's kind of like, so I'm a E. coli bacterium, and I've developed in one of my plasmids through mutations or through genetic variability, which we'll get to how that's generated in a little bit. I'm talking about once we get that variability, and it's advantageous because I can live through this antibiotic, and you're an E. coli, we get together, and guess what? There's that tube, that sex pili I was talking about, I can actually pass you that plasmid. So I can pass you that information genetically. You don't have to have been lucky enough to be born that way. You just have to meet up with another cell who actually has the right plasmid. Right? It's kind of freaky. So they can do that, like you see in the lower right. They can sit there and they can pass that genetic information back and forth. That's actually conjugation. Right? So we'll talk about. We'll talk about conjugation here in just a second, but that's what I've been referring to, conjugation. That's what they're doing. They're actually passing genes to one another. Often it's a plasmid. Okay. Yes, they asexually reproduce. There's nothing like sexual reproduction in prokaryotes. Sexual reproduction, by definition, is two different organisms combine their genetic material, right? They combine their gametes. Everybody knows the birds and the bees. I get that. And of course, sexually reproducing organisms have meiosis with crossing over and all that stuff, and that creates variability for reasons that you should remember from Bio 1. But that doesn't happen to prokaryotes. They don't have any meiosis. When they divide like mitosis. They divide, boom, equal. Like if you took something to a Xerox machine and made a copy of it, or took something to a copier of some type. All right, but so how do they get variability? And so if you're thinking, well, do we need to talk about this? Well, yeah, they wouldn't be here without it. The world has changed, the environment has changed, the earth has changed, and just like every other organism out there, they've had to adapt to that. And the only way they adapt to it is because some of them have this advantageous variation. See Darwin. Go back to Darwin. It works on these organisms the same way. So let's talk about where some of these variability come from. Mutations, right? Mutations, actual like point mutations, actually changes to the DNA. They can arise spontaneously in bacteria. Radiation can cause them. They can arise spontaneously in our cells. We hope they don't. But radiation, ultraviolet light, we talked about UV light and the ozone and protective for us. Well, in bacterial cells, that can cause a genetic mutation, just like in your cells, it can cause a mutation. Chemicals respond just like us, right? That can cause an actual genetic mutation to occur. Now, if a mutation occurs in your cells, okay, in your genetics of your cells, it could be silent. But remember we said that mutations are something you really don't want to count on for variability, because oftentimes mutations, especially those that affect the phenotype that show up, generally are bad. They're generally detrimental. So how do prokaryotes get away with it? Well, one of the ways they get away with it is by those plasmids I just mentioned, right? So this mutation, once it arises, you don't have to hope as a bacterial cell or a prokaryote to be lucky enough to be radiated or treated with chemicals and get the right mutation. You can get it from somebody else, like these two. So like the one up above might be actually getting the mutation for the one down below to have those fimbriae, those things to stick to. And that could actually cause the one up above to actually now be disease causing. Because whereas before it couldn't stick to anything, now it can. All right, so we'll talk some more about that too. That's how E. coli, the bad things about E. coli come around. So Staph aureus that we talked about, MRSA, methicillin resistant, we believe that's how, in many cases, that's how it spreads so quickly through the healthcare industries, because of a mutation to a plasmid, and then they passed it through back and forth between each other. And they're like rotten little school kids or something like that. Again, there's no meiosis, I got to remind you. Right? So it's horizontal gene transfer. All right, so we'll come back to conjugation in a second, but let's do a little review. Let's talk about some genetic variability, where else they can get it. So mutations work, and we're actually going to come back to that and talk a little bit more. Do I want to do it now? Hold on a second. Yeah, let me do it now. I can at least use it and just reinforce it. All right. The mutations that arise spontaneously, most of the times they're going to be bad for organisms, right? We talked about that. They're bad for prokaryotic cells, just like you and I. They're most of the time, especially since prokaryotic cells don't have introns, they don't have the part of the DNA that's not read. Many of them don't. When a mutation occurs, it's felt right away, and it is usually bad. So most of those critters, most of those mutations are going to end up as death or a less fit organism. And how do they get around it? They reproduce a lot. Remember we were talking about that with E. coli, and one day how many E. coli cells come from one E. coli through asexual reproduction? Like 2 times, was it 10 to the 10th or 10 to the 18th or something like that? So what you do is you overwhelm the odds. If you tell me, okay, listen, 1 out of a million, 1 out of a billion. 1 out of a billion mutations is going to be beneficial. 1 out of a billion. Well, for us, you're like, geez, we can't wait around for that beneficial mutation. But an E. coli who's making billions a day can. 1 out of a billion, sure, I'll make 20 billion. So do the odds. Some of them are going to survive. And once they're there and they can survive because of that genetic mutation, they just randomly happen to get. It's like that roll of the dice that we talked about. Guess what? They're the fittest. And if that mutation occurs in the plasmid, oh my God, then the barn's coming down, right? Barn's on fire because they're going to start passing that between each other. Oh, okay, so let's look at transformation. I said that's a review. The reason why that's a review is because you talked about it with Griffith. He was the guy that did the work with the mice, injecting the mice with the different strains, and he was able to show that a molecule, DNA, was contained information, because that's how the strain that wouldn't kill the mouse became deadly and actually did kill the mouse. And so what was happening here is the stuff that was alive, the strain that was alive that was not deadly to the mouse, was actually incorporating the DNA, taking up the DNA from the strain that was dead. So there was a dead strain and a live strain of the same organism. See, among prokaryotes, we use the word strain. Strain's kind of like a breed, maybe, if you talk about dogs. Same species, but maybe, you know, so there's a virulent strain, the one that causes disease, and the other one was not. So how do you cause disease or not? Well, maybe you form a slime layer that protects you from an immune system, or maybe you produce a toxin with a plasmid or something that's toxic to your host or makes them sick or something like that. But once you got that, if you die, say, for example, your DNA doesn't disappear necessarily, right? That DNA and chunks of that DNA could be sent off into the environment, fragments of it, and the other bacterial cell, the other prokaryotic cell, will take it in and actually incorporate into its own DNA. Now it's got that information into its main genome, right? Or maybe even to a plasmid. That's natural transformation. And that kind of neat. So what we're basically saying is the DNA in the dead cells picked up from another live cell, and now you have that genetic information that might make you virulent. I think that'd be cool if we could do that. It'd be cool if we could photosynthesize it. It'd be cool if we could pick up DNA from, I'd pick up bird DNA so I could fly. Then I want to give it back, because then I want maybe like shark DNA for a while to be what it's like to be a shark or something like that. This is what we do in the laboratory, by the way, with E. coli, the non-virulent E. coli. We actually inject genetic information in them to get them, use them as little factories to make stuff for us. Like if you injected the genetic information in E. coli for how to produce insulin, E. coli don't use insulin, but we do. And then you grow up a bunch of E. coli, you basically are engaged in this thing called molecular cloning, because you're making copies and copies and copies of the DNA. So the E. coli just becomes the little factory. The information inserted with the insulin is what we want. E. coli make insulin, make insulin, make insulin while they're reading their own DNA. So unwittingly they make insulin, if you will, and then we use that, of course, for medical purposes. So we can program them. Genetically modified organisms, kind of what that is, to be honest. Oh, too far. Wait a minute, go back. Hold on a minute here. Oh, there we go. Got a little lag. All right, there we go. This is conjugation. So I've talked about this. So imagine a little round thing is a plasmid. In this case, we have a title for them. We're going to call it a cells. If you have the plasmid, you're F positive. Without the plasmid, you're F minus. The plasmid might contain information on how to be resistant to something, you know, or produce that toxin that I've talked about. So the one cell has it, the other one doesn't. They form, one of them forms a conduit, that sex pili. So in this picture here, again, maybe it's for them fimbriate or adhere to something. Now, one thing, don't quickly run through this picture. Notice the way, I love this. Notice the way the plasmid is being copied. Only one strand of the DNA moves from the donor cell to the recipient cell, right? So the DNA is unzipping in the left, giving the other cell one strand. Then the strand that was left in the donor cell, the other side gets copied, and then what it's being passed to the recipient cell is the template. Remember, that's the thing about DNA, it's a template. If you give it one side, it could make the other side. So and this ensures good copy. Same reason why DNA replication is semi-conservative. Remember, DNA replication works by unzipping the DNA, the ladder, right? And now you got 4 strands, and then or matching it up to end up with 4 strands. So, like I always say, I tell my students, I'm not an artist, but if you give me a piece of tracing paper over a picture, I can do a fairly decent job, right? Much better than I could do freehand. Same thing with DNA. Fewer mistakes when you copy DNA that way. Same thing you copy plasmid. All right, so they can pass those things back and forth. Oh, viruses. This is the last one. This is what we call transduction. This is DNA information swapped between bacterial cells through the use of a vector. Now, we're going to talk more about vectors. Vectors are things that transmit something. Like mosquitoes would be a vector for lots of diseases. Malaria comes to mind real quick. The assassin bug is a vector for something called Chagas disease, which we'll talk about. These viruses can be vectors for genetic information. And these are a particularly cool kind of virus. These are actually called bacteriophages, and which literally means phage to eat. These are bacteria eaters, bacteria killers, basically is what they're saying. They look like little spaceships, don't they? And here's a bunch of ones in this picture down below that are attacking a cell. They're all around it. Now, viruses are these weird things. They're not alive or dead. Well, I don't want to really get off on a tangent. Your book's got a chapter about them, but of course, I don't go into it. I don't have quite the time. But the reason why they're not really alive is they can't do all the characteristics of life. For one thing, they can't really reproduce. They can carry around DNA, but they can't do anything with it. They need a host cell, like in the picture in the upper right, an E. coli. They inject their viral DNA. They inject the DNA, just like a virus does on your computer. Just kind of like we do with transformation, they inject it into the E. coli, and the E. coli makes more viral particles. So it's a cycle. That's what we call viral infections, a cycle. Don't worry too much about the detail here, right? At the top is the phage, the bacteriophage, the virus is injecting a piece of DNA. It causes the DNA of the donor cell, or I'm sorry, the recipient, right, the E. coli, let's say, to disintegrate and fall apart. And what it does is it actually uses the ribosomes. It hijacks the genetic material, more specifically the ribosomes, in that cell to make more viral particles. So the ribosomes are cranking out proteins. They don't know what they're making. And the information, the viral DNA, causes them to make viral particles. And then the cells rupture. That's usually what causes the problems with people with viral infections, the lysing stage. Like folks with malaria, that's when they get the fevers. Actually, in that case, that's a parasite that's causing the cells to erupt. But then that phage, that bacteriophage, goes to another cell, and oftentimes when those viral particles are being formed in the head, there you see the little blue squiggly. That's more viral DNA, but it's kind of sloppy. And sometimes they'll stick in bacterial DNA. So basically what it is the virus acts like the genetic mix-up. It moves the genetic material around. Now, again, how often does this result in something bad for the recipient cell? Probably a lot, but you got a lot of numbers here too, again, right? It's all about the numbers. If you're making a lot of offspring, you can experiment a lot with moving the DNA around. Okay, how about nutrition and metabolism? How do these guys make a living? Now, before you go crazy looking at this picture or this slide, the next slide, which is not in yours, but there it is. I've taken away the amplifying information, right? I'm giving you examples in each of these, but basically, what we're talking about is in organisms in general, whether they're talking about prokaryotes or protists or any living organisms on this planet, you need two things. You need a source of carbon and a source of energy. All right, source of carbon and a source of energy. You need the source of carbon because we're a carbon-based life form. You need a source of carbon because you use that to build up your structure of your molecules. You need a source of energy because that's an energy-demanding reaction. So we can break down your source of carbon through something that's inorganic, like carbon dioxide, which is what plants do, right? Or we can break that down into organisms who get their carbon from other organic compounds. Like, for example, you and I are that way. We get our carbon from eating things. See the picture of us right there? We're an oxidizer, and that's the energy that we use. So the energy is either light or the oxidation of certain materials. Now, look through each of these and look at some of the examples that you got and read through that. But Bottom line is I want you to be able to tell me at this level what the difference between those two. So if you have those notes and you want to circle that, I'm not going to get into the details too much. We are going to talk about some of these organisms later on, like, for example, actually, we already mentioned the Halomonas titantia that's using iron and oxidizes iron. It's just amazing. Just floors me. But here's the big thing, right? I want you to look at carbon source and energy source and be able to use these words. So something that uses light as a source of energy and carbon dioxide as a source of food is considered a photoautotroph. So plants would be that. You can also get energy from oxidation. You can do that inorganically and be a chemoautotroph. You could do it through the oxidation process, either through other organic materials as your source of energy and your source of carbon, or light as a source of energy. Now, notice though the prokaryotes. Prokaryotes are the only ones that can do these really bizarre kinds of ways. Like the ones on the one on the left, the light and the carbon dioxide, that's plants, right? There's plants. That's us. But what we're saying here is these organisms use carbon dioxide and they can oxidize inorganic materials. So they get their energy not from organic materials like we do, but inorganic materials. And these guys get their energy from light. Okay? So that's what the other ones are examples. And it's important to look through the examples, but I'm not going to test you specifically on the examples. All right, in terms of oxygen, some are obligate anaerobes. Obligate aerobes, so they require oxygen. That'd be us. Some are obligate anaerobes. We talked about that the other day. They might have been some of the ones that were avoiding oxygen levels when they rose on Earth back, 2 billion years ago or something like that. These are the organisms many times that are fermenters. They're those engaged in anaerobic respiration. And then there are those that can, they're the variable ones. They're facultative anaerobes. Facultative anaerobes can live with or without oxygen. Many bacteria, fortunately for living organisms on the planet, are good nitrogen fixers. And nitrogen fixing, if you look in that chart to the right, the part in the red, that's nitrogen fixing. Nitrogen fixing is taking atmospheric nitrogen and converting it into something, some chemical compound, which then can be used by, for example, plants in this case. Remember I said this is fertilizer. Plants can't fix atmospheric nitrogen. It's 70% of the air, but they need, in many cases, these bacteria that can do that, that can create these ammonia, then nitrites, and then nitrates, and turn those into plants. Or not turn those into plants. And plants can use those as important parts for their own life. You need nitrogen to survive on. It's one of the major components of DNA, right? So there's a lot of prokaryotic organisms that can do that. There's also a lot of prokaryotic organisms that work the other end of the spectrum. So they denitrify stuff. So they turn from nitrates to nitrites and then back into atmospheric nitrogen. So the amount of nitrogen, you don't want too much, you don't want too little. Too much nitrogen can cause a problem in the environment. Too little, not enough for the plants. You also got animals decaying and plants decaying, and they're decaying and producing nitrogen compounds. So it's all a big balance in the ecosystem. And bacteria and prokaryotic cells are a big part of that. The other day when we looked under the microscope, we saw Anabaena. This is Anabaena, a little better picture, but again, it's kind of like the ones we saw were dyed more of a blue-green color. Anabaena is a filamentous, a long filament of cells, and there are two types of cells. I think Jessie actually noted that when she was drawing a picture of the Anabaena. I remember she said that she noticed there was a different type. There's the photosynthetic ones, the green ones, and the other ones, the heterocysts, they do nitrogen fixation. Look at that. Anabaena is really beneficial in two ways, right? Anabaena is a photosynthesizer, so oxygen levels, good, and nitrogen fixer. So this would have been a very good critter to be one of the first critters to move out into land, right? Producing oxygen, that's good for eukaryotes, and then the nitrogen fixation, right? Now, if you don't brush your teeth, you're going to get a biofilm. That's what plaque is on your teeth. That's what that picture is. Is that a piece of tooth, I believe? or tooth material, and the blue is all the Streptococcus bacterial growing together in this big biofilm. And the reason why it's a problem for your tooth is Streptococcus then will begin to anaerobically respire and form sugar, or from sugar, and produces lactic acid. Right. So when you ferment things, oftentimes, you get lactic acid, or you get a metabolic compound. Sometimes it's not good. And in this particular case, the Streptococcus, when it ferments, the lactic acid is bad because it eats through your tooth enamel. But biofilms are kind of like these colonies that we were talking about, where they live together kind of as cooperatively. It just makes them more efficient. We start to see this metabolic cooperation. Not quite multicellular, though. Oh, gosh, 10,000 or more species, probably higher than that. We used to classify them on very simple things, like we've talked about, whether they could photosynthesize or not, or move, whether they were filaments grew together, whether they're unicellular. And then finally, a lot of times, we tend to classify things based on what they mean to us. So importance is human pathogens. But the problem with that is very little of that may have to do with genetic relatedness. So lately, we use a lot of those DNA analysis we talked about, right? Amino acid sequences, which is going to give the DNA, comparing the two ladders of the DNA with base pairing, and then looking at the whole genome. And what we've come up with is 3 domains, of course. Eukarya, that's where we are. We're not there yet. But domain archaean bacteria, the 2 groups of prokaryotes. And we'll talk about some of the archaeans, and we'll get in a little bit more with the bacteria. But the red exclamation part is because I want you to notice when you look at that eukaryotes, us included in the fungus, everybody else, we're more closely related actually to the archaean prokaryotes, the ones that are considered more ancient, archaic, than we are actually to the other types of bacteria, or the other types of prokaryotes. I think that's kind of neat. Because when I talk to you about the archaeans, especially a lot of them, you can say, wow, those are some pretty hardy type organisms. You wouldn't think necessarily that they would be necessarily related to us. Now, they're similar to us in a few ways. Focus on the arrows in this chart, really. Don't worry about the other stuff so much, but do read through it. Bacteria and archaea prokaryote nucleus, so they're different than us in that way. Archaeans don't have peptidoglycan. We talked about peptidoglycan, but it was only among bacteria, not among archaeans. The lipids are different, and we'll get to that a little bit later. Actually, next slide, but we'll get to that in just a moment. As I mentioned, the introns, the intervals, you don't find very many of them in bacteria. You usually don't find any. Archaean have in some of the genes. We have it in a lot of our genes. That's the reason for processing. It's the reason for messenger RNA processing, right? Got to get rid of the introns, the intervals. Now, here's the part that maybe throws you off when you think about who we're related to. Archaeans tend to, many archaeans tend to be the extremophiles. Not all of them, but many, those organisms that can live in very extreme environments, like temperature, salinity, the extremophiles. So a lot of archaeans are extremophiles, grow at temperatures like beyond the boiling point, beyond 100 degrees Celsius. So if you think, wow, that's probably some of the first critters that were on the planet. So it kind of, when you look at that, what we're saying is when those first critters evolved on the planet, the universal common ancestor was probably archaean-like. It probably could stand some pretty harsh conditions. And then what happened is that, that split into the bacteria. They came off first, and then the archaeans split into the eukarya. So the bacteria were the first to split off from the bacteria, or from the archaean main line. So maybe the archaeans were some of the first critters on the planet. Oh, one of the reasons they might have been the first critters on the planet too, or the one reason they can survive these really extreme conditions, because they make some super duper plasma membranes. I got a couple of pictures here. If you look at the one in the upper right, that's a little simpler first, and then I'll add more detail to it with the one on the bottom. Members in the domain bacteria and eukarya, us in the bacteria prokaryotes, we have the typical phospholipids. But in the archaean, they have phospholipids that are branched and they're connected to each other. And that actually causes their plasma membranes to be layered. The bottom line is that it causes them to be much more durable. The plasma membrane of an archaean is much stronger, much more resistant to environmental conditions. That's why they can be the extremophiles. Another reason has to do with the type of chemical bond that we find in bacteria and eukaryotic phospholipids. It's called an ester bond. Now, if you've had chemistry, maybe that means something to you, but for our purposes, the archaeans have what's called an ether bond, and ether bonds are more resistant to hydrolysis. So again, the idea is we get this stacking of these phospholipids. You can see what I mean over on the right, the tetraether. So that's what an archaean bilayer looks like. It's more like a monolayer, because what they're doing is they're kind of laying on top of one another. So they're connected across this way, and they're also connected across this way. They're branching together. They're sticking to one another, so they form this very, very resistant layer. So with the ether bond and the resistance of the way they form those monolayers, they can actually allow these organisms to survive in extremely hostile environments, very warm, or salinity. What else we talk about? Radiation. I mean, these are some pretty heavy-duty critters. That's why when folks talk about life on other planets, I'm like, it doesn't have to be perfect. You need some few basic things, but it doesn't have to be like Paradise Planet Earth kind of thing. All right, let me run through some of the major bacterial groups. So we have the archaeans. Those, as I said, are the extremophiles. And among the archaeans, the three big ones that we think about are the methanogens, the halophiles, and the thermophiles. The methanogens, they're the ones that actually produce methane. So they use carbon dioxide, they oxidize hydrogen, and that produces methane. That's what's given off in swamps. That's the smell that you smell going through a swamp, is the methane gas. Remember I said in class that that's significant, because presence of methane, or on exoplanets, other planetary bodies, might indicate at least maybe domain archaean type organisms. These are obligate anaerobes, so they're going to live down in the mud of these swamps. They don't survive in oxygen. So those mud of swamps in the bottom of lakes can become very low in oxygen. The halophiles, like you see on the right, the ones that make these really pictures, they live very salty places. And the thermophiles can take it if it's very hot. Their optimum temperature, by the way, not upper limit, is about 60 to 80 degrees Celsius. Body temp in the human is 37, just for comparison. These are smoker vents, those hydrothermal vents we talked about in chapter 26. I think we discussed, no, 25. Find a lot of those thermophiles in those places. You can find a lot around these hot springs too, in these hot and very salty places. So there are thermophiles and halophiles in the same organism. Now switching over to the bacteria, we have a clade of bacteria called the proteobacteria. These are all gram-negative, so they all look red under a microscope. And we separate these into some major groups, a half dozen or so. How many do I have listed? 3, 5, 6, 7, 8, 9 major groups we got listed here. The first 5 groups go by the Greek letters alpha, beta, gamma. Then there's delta and epsilon. We'll get to the next slide. Alpha bacteria are very often photosynthetic parasites or symbionts. They're very often associated with the eukaryotic host. Remember I mentioned alpha proteobacteria might have been where mitochondria came from. There's a picture of some alpha bacteria, the rhizobium, inside the root cell of a bean cell. Many of these can fix atmospheric nitrogen, so that's why you might find something in the root cell of a legume, of a bean, because it's fixing nitrogen. Beta bacteria can do that as well. They can also fix nitrogen. Very often found in soil. Maybe some of the bacteria we found in some of our plates from the soil have beta bacteria. One not so wonderful beta bacteria is Neisseria gonorrhoeae over on the right-hand side, or gonorrhoeae. It's a diplococcus, and it's the organism that causes gonorrhea. All right, so in each group, there's the good and the bad. The gamma bacteria are the sulfur bacteria. They can actually use sulfur as part of their metabolic process. A lot of these are pathogens. Remember I said gram-positive, gram-negative. We might want to know if it's gram-negative, because many gram-negatives are pathogens, disease-causing organisms. And in the case of the gamma, There are the E. coli that can be non-pathogenic or pathogenic. The one that causes Legionnaire's disease, Salmonella, and then one that causes cholera, Vibrio cholerae. These are all sulfur bacteria, or gamma bacteria, excuse me. The delta bacteria can move along by those slimy threads that I was telling you. For years, these were confused with fungus. If you look at the picture in the upper right, it kind of looks like it's going to look like some of the fungus we talk about. This is we call these things myxobacterium, and the myxo part is a root that actually refers to a fungus. So that's the origin of why it says that. They're not fungus. They're still prokaryotic organisms. Delovibrio is a neat one. In the picture on the right, Pseudomonas is being attacked by Delovibrio. Delovibrio is a predator. It can actually swim pretty quick, up to 220 miles an hour scale-wise. And then what it does is it attaches to Pseudomonas, and then it spins like a drill. Zzz, and it spins through until it punches through and then destroys the organism. Epsilon bacteria, many of these are pathogenic to humans, and for some folks this has actually been helpful to find this out, because we found out that some folks suffer from stomach ulcers caused by bacterium, Heliobacter pylori. So with just a few simple antibiotics, these folks have been relieved of their symptoms. I think I'll probably 15 minutes, so we'll stop a quarter after and then go and talk about the exam. There are the chlamydias, which are dependent upon animal host cells. So these are going to be parasites. Trichomonas is the most common cause of blindness worldwide. So these diseases many times are sexually transmitted, but can have far-reaching effects. Non-gonococcal urethritis, NGU or NSU, that's also caused by a chlamydia organism. Most common sexually transmitted disease in the United States, and silent. Look at the picture down below. I got to go shut the door for a second here. Look at the picture down below. Whereas the rates of gonorrhea, is that the yellow? Yep. And syphilis have gone down. Look how chlamydia has gone up. What does it mean when something's a silent STD? What do we mean by a silent? That explains why the rates have gone up, right? What's a silent, or anything that we consider silent? We talked about silent mutations too, or you did in Bio 1. See, the nefarious aspect of these organisms that are silent, like this one, is silent STDs don't show symptoms, so therefore they're being spread because you don't know you're symptomatic. That's the problem with that. The spirochetes, we look in the picture, they're obviously the spiral-shaped organisms. Many of those are free-living, but there's a few that we're more familiar with. The one that causes syphilis is the spirochete, Tryptonema pallidum. And Borrelia burgdorferi is the organism that you see in the picture that comes from the Lyme, or comes from the deer tick that causes Lyme disease. There's the deer tick. Now, we don't have a real problem with deer ticks here in Florida. There are deer ticks, obviously, but the deer ticks are more prevalent up in the Midwest region. So most of the ticks stand around here, at least all the ones I've seen have just been your common, brown dog tick. The cyanobacteria that we introduced ourselves to the other day were important because they were the photosynthesizers. They're the only ones that photosynthesize like eukaryotes, which is good. They produce oxygen, but sometimes they can have these blooms, and that's where you get that pea green colored lake that you see there on the right. Look at the difference between that one and the one just across the little berm. Nutrients cause this. See, these are photosynthesizers, so they're kind of like plants, like Oscillatoria there in the picture, and Anabina. So when you dump a bunch of nutrients like fertilizers, phosphates, of nitrogen into waters, that can cause algae blooms. And while that's good for the algae sucks up a lot of the oxygen too, because they also respire. The gram-positives, there are colonial versions, the actomycetes like streptomycete, which is a very important source for antibiotics. Now again, let me remind you, when we talk about colonial prokaryotic cells are considered unicellular critters. We don't get to this multicellular stuff until we really start talking about, especially into the animals. But solitary, there is bacillus, the anthrax, the one that causes botulism, Clostridium botulinum, produces endospores, produces an endotoxin that is so fatal a gram can kill 1 million humans. Nasty stuff. Staph and strep are both examples of gram-positive solitary prokaryotes too. All right, what do they do good? Obviously, we'll talk about the bad in just a second, the disease, but they're good for chemical recycling. Prokaryotes are good decomposers. They break down dead organisms, waste products. They can photosynthesize. They're one of the only organisms that can break down this substance called lignin. Lignin is a very complex chemical structure. I've got its, this is its chemical structure of lignin. Lignin is found in wood, and lignin is very stiff. It's a very hard chemical structure. What helps trees stand up? It's in addition to the cellulose in their cell walls. But lignin is very hard to break down. Only a few organisms, some fungus and bacteria, can break down lignin. And if it couldn't be broken down, that's a lot of carbon loss to the environment, right? There's a lot of carbon atoms in there. Remember, carbon is the foundational element. You learned that in Bio 1. So that's a lot of decomposers recycle that carbon. Many prokaryotes engage in symbiotic relationships. I've gone over this with you before in class. So there's a little chart to help you. The only thing I would add is parasitism also includes the symbiotic relationship of predation. One gains a benefit, one does not. So there are prokaryotes that engage in those kinds of relationships. Mutualism, for example, your gut microbiota, the intestinal flora of your gut. You see in the picture on the right, there's what, 10 to the, oh, my glasses, 14 microorganisms living throughout in your stomach, increasing in concentration down through your intestines, and they're all very important for proper digestion of your food. 1,000 species, 10 to the 14th. I mean, obviously, those are important. So not all bacteria are bad. You know that some folks are suffering from gastric disturbance is because of some imbalance in their intestinal microflora. We can hijack them, get them to make insulin. We can get them to make stuff like PHAA, polyhydroxyl alkanonate, which is a plastic, a biodegradable plastic. That's what that stuff is inside those little organisms in the picture. We do what we do with the genetics. We program them to make this stuff genetically. And they don't need it, and then we just, I don't kill them off and take it, whatever. The guy down below is not got a very enviable job. He's spraying this mixture on oil-covered rocks because there are plenty of microorganisms out there that feed on oil. Remember, oil gets into the ocean by us dumping it. But it also gets into the ocean because of natural leakage. Throughout history then, throughout evolution, organisms have evolved to eat that. Oil is hydrocarbons. It's a source of energy. So this guy is actually spraying the rocks in Prince William Sound, where the Exxon Valdez ran aground back, late 80s, and dumped a bunch of oil. And what is in that spray is these prokaryotes who dissolve oil, who eat oil. It's not a perfect solution. For example, even in Prince William Sound today, they say all you have to do is dig down a few inches and you'll find the oil. It looks good on the surface, but the oil's still there. Same thing in the Gulf. Even though we can't see the oil that spilled back in the summer of 2010 like we used to on the pictures or the news, it's still there. That dispersant they sprayed just makes the oil smaller. It doesn't make it go away. They think there might be clouds of oily water moving through the ocean. All right, but of course there are the ones that we hear about that are disease-causing. In the early 20th century, about 20% of children died before they were 5, very often from some sort of microbial pathogen. A lot of it has to do with our misunderstanding or our lack of knowledge of hygiene. You remember the olden days, whenever that might have been. It was before my time, thank goodness. Your chamber pot that was used throughout the night, you just threw it out the window, onto the street down below. So hopefully, that. They say that's one of the reasons why, what is it now? Men always typically are supposed to walk on one side of a woman versus the other, and that came from the idea that the man would walk closer to the building and he would get the stuff thrown on him from above versus the woman. All right, but now we know, of course, and we have much better sanitary provisions, but the organism on the right, the Mycobacterium tuberculosis, you've heard about it, obviously. Now there are multi-drug resistant strains for the same reason we have MRSA. A lot of these organisms, like tuberculosis, for example, secrete, I think it's tuberculosis who secretes an exotoxin. Now, the problem with the exotoxin is the organism doesn't have to be alive. So it secretes the exotoxin, and then you can actually, like maybe into a food item that you eat. The organism dies, and this exotoxin is still there. That can be sick. Endotoxins, those lipopolysaccharides on the outside of gram-negative bacteria, those little green things, those can be released when the bacteria die, their cell walls break down, and those things can cause the symptom can cause disease. And then, of course, when it comes to disease, once again, their ability to switch back and forth with horizontal gene transfer, that was Griffith's work. That's how we can get pathogenic ones, like the E. coli strain. E. coli strain, the O157H7, that's the pathogenic one. You have E. coli in your gut. All warm-blooded vertebrates do. It's part of your natural microbiota, the flora. But you don't have the pathogenic ones, or you'd be sick. The one that we get in the food is the pathogenic. And the difference in them is those fimbriae that I've been showing you throughout. The ones without the fimbriae can attach, and they don't cause disease, but the ones with... and the one, once you have the plasmid for making fimbriae, you can pass it horizontally between your buddies. Against like nasty little middle schoolers, right? Passing a note around. Okay, that's all for the lecture on 27, and I finished just about on time. Gives me about 15 minutes before class officially ends. But does anybody have any questions for me about the exam before we get, any general types of questions? I can't sit here and tell you, well, yeah, I was just going to say, I can't sit here and tell you specifics like that, because I would just be doing nothing but basically horsing the exam to basically make sure you know page 4, which is not learning at all. But I would expect you to know some of those people. All right, and what I might do is not say, okay, name all the scientists who were involved in, this science of evolution from Aristotle all the way through Darwin, but I might use these folks' name in a question, and you have to know kind of what they were involved in. I might say, Hutton's theory of geology. Well, in that case, you don't necessarily have to remember Hutton was a geologist, but you have to kind of remember what he said. He said the Earth was very old and all that kind of stuff, right? You see what I mean? Not to say that details aren't important in your exam, but I'm not someone who's gonna, really stick you with a bunch of these minute little details, like who said this and who did that. Let me move you guys to the other computer so I can get a better view. Just 20, yeah, 22 through 26, including 25. That's right. So the exam is scheduled for the entire hour and a half. You, of course, can take the entire hour and a half. I would encourage you to. I mentioned, you can use your notes the last 15 minutes of class. Now, if that means you get done in 20 minutes and you don't want to sit around and wait, because you got to wait till the last 15 minutes of class, and you think you did well enough, then help yourself. You don't have to stick around. You can leave. But if you do want to be able to use your notes, you're going to have to sit there patiently until the last 15 minutes of class. I would encourage you all to stick around, at least on this first exam, just so you kind of know what kind of questions I ask and feel comfortable. I would encourage you all to take the entire time. You may not need the entire time, and you may find later on you don't need the entire time. But again, if you leave before the 15 minutes, you'll forego the opportunity to use your notes. Yeah, I can talk about that. You got 2 essay type questions. One is a more traditional essay, and in that, okay, here's what I do with my essays. Let me see if I can copy and paste this. And I'm probably going to have to read this. I'm going to read it in case it doesn't show up on the chat. In no less than 250 words. I always have a word required in my essays because some of you would like to give me bullet points, and bullet points aren't answering an essay question. Bang, bang, bang with facts. I want it to be cohesive because I want you to be able to demonstrate to me that you understand what we're talking about. So discuss evolution, the principles of Darwin's mechanism of natural selection. All right, so I want you to tell me about evolution, and I want you to tell me about the mechanisms of evolution. If you remember, it wasn't just Darwin's, there were others. So it's when you're thinking about how I would be able to answer this question, keep that in mind, that I'm probably going to make you compare those different ones. And then I have a part where I say your answer should include discussion regarding the following. Now, I'm not going to give you the following, but it's a series of 1, 2, 3, 4, 5, 6, 7, 8 key points that I want you to touch on. These aren't meant to be suggestions. These are meant to say, in order to wow me, and there's a rubric, you'll see it after the exam. In order to wow me and max out on the rubric, you're going to have to touch on each one of these topics to a sufficient level. So if you're trying to think, well, what could he mean by these topics? Well, if I was going to discuss with you evolution, I'd want you to be able to tell me the difference between, like, say, drift, gene flow, the difference between that and natural selection, why one is adaptive and why one isn't, how does the founder effect and bottlenecking fit into all of that? What you should definitely know Darwin's major points of his theory, right? That more are possibly born that can hope to survive, resources are limited, so on and so forth. Make sure you can discuss these things adequately. 250 words. Now, that's not a lot, right? I try not to just go crazy with these huge essays, because I find people write the same thing over and over again. But I do expect you to give me a adequately written essay. In other words, I'm not going to nail you on grammar a lot. If it's to the point where I can't understand what you're saying, or if it's text speech, for God's sakes, please don't give me text speech. Don't give me the shortened version how you text when you're texting. I do it too, but obviously on an exam you can't do that. Think about the way when I grade that exam, I got to sit at home or wherever I am, and you got to convince me through the written word that you understand the concepts to the level that satisfy me that you can say that you mastered proficiency with this concept. And out of all those chapters we discussed, 22 through 26, one of the biggest things I need you to understand is how species change and why they change that way. Does that help? And the rubric is going to grade, well, first of all, it's real simple. Yeah, I can do that. The rubric's real simple. First of all, the first thing it looks at is how long is your answer. I'll nail you if you're short. Even if you get all the right information, I want you to discuss it fully. I doubt you'll get all the right information in less than 250 words, to be honest. I give you sort of a gimme on spelling and punctuation, as long as it's not just vomiting on the page and horribly written and just a collection of random thoughts. I'm going to give you, the full max, and then I grade you on whether you use the right words, whether you define the words properly, whether you even define the words. So if you're describing the bottleneck effect and you go through all this description, but you never say bottleneck effect or say that it's inbreeding or can lead to that or maladaptive, I'm missing the terminology. And then the last part of the rubric basically is, have you demonstrated to me, and at what level did you demonstrate to me that you are proficient with this information, okay? Oh, and Tatiana, good. I'm glad you brought that up as well, because the other essay, it's not really an essay because it's well, it is kind of, is you will have to do a Hardy-Weinberg problem, just one. And I'm not going to throw you a curve. I'm going to be nice. I'm not going to give you ones like we're in the assignment where like the pink flowers and the red flowers. I'm going to give you a very simple one. I'm going to say X amount of critters, X amount of them are recessive, right? And then I might go through and ask you, then you're going to do your calculations. Remember that step, those steps I gave you. Identify Q squared, solve for Q squared, solve for Q, solve for P, square P, 2PQ, right? Do your checksum, everything adds up the way it's supposed to, and then use those proportions to answer the questions. Now, this was on the was on the Hardy-Weinberg assignment. One thing that I think it's important that you guys keep in mind is the Hardy-Weinberg is at equilibrium. That because the numbers change, we're saying that something's in disequilibrium. So you should definitely know what are the conditions of the Hardy-Weinberg that cause those equilibriums to change, like gene flow and drift, right? Remember, only one of those conditions was natural selection. So let me ask you a question. If you were studying a population a certain flowering plant, right? And you calculated these values, these Hardy-Weinberg values, and then for all the phenotypes, and you got heterozygotes 48%, okay, and then you come back in, let's say, I don't know, 20 years, and now you calculate the heterozygotes are now 46%. Based on the change that you see there, so this has been several generations down the road, could you argue for or against that the population is still in Hardy-Weinberg equilibrium? After 20 years, X number of generations, with the numbers I gave you, heterozygotes were 48%, now they're 46. Can you say, with fair degree of confidence that the group of flowering plants is in equilibrium? Now, don't get too fixated on the 48 and 46 and am I supposed to know something about the difference in those numbers? I'm just using those two numbers as comparison. I could say, all right, the proportion was X, and when you came back in a few years, it was just a little less than X. Or maybe it's just a little bit more than X. It wasn't exactly X. I mean, my goodness, what are the odds you get exactly the same number? What if I asked that question on an exam? On an exam, I said, all right, here's the situation. Do you, would you indicate that these organisms are in equilibrium or not? Remember that thing we did with the beans? We found out that that wasn't in equilibrium. Those numbers changed dramatically. I don't know. I don't get that. Probably yes, because the environment may change. Probably no. I don't understand. Someone help Suzanne out. Jesse? Janae? Madison, come on. What does that mean? So, I go out there and I do the proportions. Okay, I go out there and I do the proportions of the Hardy-Weinberg. I calculate all those values. I come back in 20 years. I calculate them, and they're just a little bit different. They're just off by just a few fractions of a percentage point, or even just a couple of percentage points. Okay, you can argue both ways, right? But is that difference that you see, 48 to 47 to 46%, 48 to 50%, is that enough to indicate to you convincingly that the population is out of equilibrium? And if it is out of equilibrium, what caused it to be out of equilibrium? Or are those numbers close enough that it's probably still in equilibrium? And what does that say? Yeah, they're still in equilibrium, right? So what does that mean if they're still in equilibrium in terms of someone says, well, Dr. Whatever, Dr. X, based on your results, would you suggest that that population of flowers has been evolving in the last 20 years? So if the numbers haven't changed, yeah, there's no evolution, right? Remember, don't confuse evolution with natural selection. One is the theory, one is the mechanism, right? Evolution can change through drift. Evolution can change through gene flow. But if the numbers don't change, evolution, no matter which way, didn't happen. So those populations in equilibrium. Evolution is not happening. Now, if the numbers were substantially different, then your answer would be different to that person. Well, yes, my numbers indicate evolution has happened. Well, did it happen by evolution through natural selection, Dr. X? Well, I don't know yet, because guess what? I haven't checked out to see if it was drift. I haven't checked out to see if it was flow. I haven't checked out to see if there's any mating issues. I haven't checked out to see whether there was any chance that allelic mutations, even though those things might not be probable. I have to check all that out first. Because if the numbers change, because let's say I was looking, now switch over from flowers for a second, let's go to fish. Fish in two ponds, right? And they were separate from one another. And then one pond flooded and a bunch of fish came in during a storm, which didn't usually happen. Then you change the numbers, but that wasn't because of natural selection. That was gene flow. All right, see what I mean? Bring a calculator. Yeah, bring a calculator. If you don't or you forget, don't freak out. I got 3 or 4 of them I can let you use, and you won't need them for very long. Know how to work those Hardy-Weinberg problems. Bang, 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 though. Practice those. Where are we in time? Oh, we're at 2:30. Okay, I'll keep the recording going. Y'all, obviously, it is the end of class, but you're welcome to stick around. Titania said something about working on a few Hardy-Weinberg problems. Yeah, whatever you want to do, it's fine with me. We can do that. But sticking with the exam for just a second, so we don't, so I don't lose track. Make sure you know how to read cladograms. You know those family charts that we made in class with the second lab. I might ask you some questions about that. Is this group monophyletic? Is this one paraphyletic? So you've been practicing that with your assignment. Make sure you understand things like when I use terms like convergent evolution or common recent ancestry or just recent ancestry. Synapomorph and plesiomorph, that goes along with those cladograms too. Pace of evolution, you should know that. The overall idea of the exam is you're going to get 16 multiple choice questions. Yay, right? Multiple choice. And then you get, what do you call those? That's two kind of fill in the blank. Not really fill in the blank, but you get, I give you some question and you have a drop down of answers to choose from. So I do give you a word bank. Okay, I give you a word bank. Sometimes not. The words in the word bank aren't used at all, so you have to really be a little more proficient than just being able to guess. Okay, there's that. We talked about that, and we talked about that. So I've touched on everything that is a major part of the exam. I didn't focus on the questions, obviously, but I've given you some idea of perhaps trying to see how you know, like I asked stuff. All right, here's a question. Start. No, it's on the computer. I'm not going to give you the answer, but like, here's what I said. Okay, you're going to categorize some statements. Is this gradualism or punctuated equilibrium? Is this gradualism or cladogenesis? Is this anagenesis, slow change, or cladogenesis, rapid change? So I'll give you an example. Leaves from living ginkgo trees. What? It doesn't matter. Who cares? You don't need to know what a ginkgo tree looks like. Leaves from living ginkgo trees look the same as 40 million year old fossils of the same species. All right, so then you got to tell me whether you think that's gradualism or that's punctuated equilibrium. All right, I'm looking at that question. I'm looking at the answer that I put. And I don't know if I put the right answer. I can think about that. Why did I say it that way? All right. Any other questions while I'm sitting here pondering this? Any other questions? Anything specific I can talk to you about? As I said, I like reviews to be led by the students because then that lets me know, you know, what you need me to talk about. So I'm not just here kind of going over the same information. I'm you know, some of this stuff is pretty straightforward, pretty easy. Do you guys need to talk about Hardy-Weinberg problem? Some of you said you did. Let me take a look at that. Why is that freaking me out? I don't know. You just come down to the room. Come down to the room, and if it's not there, I'll put a note on the door where it's going to be. It's either down in the room, probably is going to be upstairs, okay, in 216. But if you don't remember that or you walk in and you forget, then don't worry about it. 216, it's upstairs. I'll have some signs posted. How many questions on the test? So what did I say? You know, you like 16 multiple choice, a few of the fill in the blank, some of those cladograms, a couple of other fill in the blanks. So I don't know, I guess it overall ends up being 24 questions worth 100 points. Different point values. So it's kind of a little mix. Anything else? All the ones that, oh, okay. all the ones in the Hardy-Weinberg equilibrium if they're not violated. Does that make sense? Random mating, no allelic mutations, no genetic drift, no gene flow, no natural selection, they'll stay in equilibrium. If those are violated, they'll get out of equilibrium. Did that answer your question? If you guys, if you need to flip through your notes, whatever questions, this is your chance to. Somebody want to throw a Hardy-Weinberg problem up there on the board? If you have a touch pen, you can write on the board. Just some sort of population. We can practice working through it. I'm not going to do it. You guys are going to have to do it. So, or as you can tell me what you want me to put up on the board. You guys are doing the penguins on the edge of the ice thing. So everybody knows the Hardy-Weinberg. Nobody's has any problems with that at all. Let me just turn on their mic.
Yeah, so it'd be a lot easier than typing it.
Huh?
I don't know if you can hear me. Can you hear me?
I can hear you.
Because I have long nails, so typing takes me forever. But
let me turn the volume. Turn your volume down a little bit. Because it's echoing.
okay, so with the Hardy-Weinberg, the question 2 on the assignment.
Mm-hmm. Oh, yeah, Madison, of course. Yes. So, question 2 on the assignment of the Hardy-Weinberg. Read it to me. What happened? He vanished in cyberspace. Jesse, Danae, Suzanne, y'all have any questions? I'll wait here. No, this is... Okay. You have a good weekend then. No problem, Suzanne. If you have any questions, you can always email as well. Jesse, how about you? That's good. Yeah, no problem. I'll be checking my email throughout the weekend, so if you have any questions, give me a holler.